Chapter 20, Phylogenies and the History of Life. As we've seen before and going forward, there are many different living things. Some things that they all share, organisms share, are that they are all composed of one or more cells. They all carry out some form of metabolism or a series of chemical reactions. They transfer energy using ATP, and they store their genetics, their hereditary information in the form of DNA, so nucleotides. Now, how diverse is life on Earth? We know that there's about 1.5 to 2.3 million species that have been identified. But is that the total number of species? No. We think there's probably 5 million or even more species that are out there that many of which we don't even know about yet. So if there's that many species, how many individuals? Well, we think that the total number of individuals could top 100 billion or even 1 trillion individuals when we think about how many different individual organisms there are. So given this, it's very important that we are able to group organisms into a categorization so that we put them together based on characteristics that are alike. So how can we do that? Well, there's a number of different aspects that we could use. We could look at fossil records to group organisms together. We could look at morphology, which is their anatomical structures. We could look at physiology, or how different components of them work. We could look at behavioral aspects, what the individuals do. We could look at how they develop as embryos, and additionally, we could look at their actual DNA or RNA sequences to see which organisms are similar to one another. So when we look at the science of doing that kind of grouping, we see that there are a number of different fields. We have systematics, which is the study of evolutionary relationships. And then we have phylogeny, which is making guesses about the relationships between different groups of organisms. So a taxa or a taxon is a group of like individuals. So from this, we can make something that's called a phylogenetic tree or a cladogram, and we have two examples of them down here. And we can demonstrate in an image the relationships between different organisms. Now, this is called a rooted phylogenetic tree. This is an unrooted phylogenetic tree. So rooted, think of like a tree. It has an initial stalk. So this all arises from a common ancestor. And so then as changes occur and new species develop, the tree will branch and then branch further. So from this, we can see that there is a common ancestor. Now, in this unrooted phylogenetic tree, we show relationships, like how close groups of organisms are, but there's no identified beginning to the relationships. Is one better than the other? No, it's just different ways to represent the same kind of information. So let's take a little bit of a closer look at some of the components of a phylogenetic tree. So we have a diagram of one right here. This is a rooted tree. We see that there was a common ancestor. And we look at taxon or taxa, which is the plural, which are groups of living organisms, so a species, a family, a domain, etc. And they're represented in this diagram by the alphabetical letters here. So we have A, B, C, D, E. Those are 5 different taxa. And the closer they are, and if they come off a single branch, they're called sister taxa. Then we have the clade. So the clade is the actual arm that reaches up to the taxon. So that's the clade, the lines. And because we have lines, we're going to have branch points. So here's a branch point or a node. So this demonstrates where we have a division between two taxa or multiple taxa. And so we have created two different clades. So here's one clade, and then we have another clade, which then branches into subsequent clades. So where we have these branch points or nodes is where we see common ancestors, and that can be traced all the way back to an original single cell, which we don't really know the specifics on that, but we have some guesses. But further up in the tree, we're a little more certain of who the common ancestors might be. So when we look at phylogenetic trees and we start talking about how do we generate these trees, we see that the trees, they're ladder-like structures where we have if it's rooted, where we have a common ancestor, and then we get to a branch point and we have to answer questions. And those questions have to do with some kind of characteristic. And it's a yes or no question, which causes us to go one way or the other way. And that way we can start breaking apart different groups. So when we start to look at the animal kingdom, we see that, and then we branch from invertebrates and vertebrates. As we start going down the vertebrate phylogenetic tree, one of our first questions are, do we have a hinged jaw in the organisms? No. Then we see that we have this group. Yes. We go on to the fishes. Then we have another question. Do we see legs? And we say no. We say yes. I'm sorry, here's the hinged jaw. My apologies. So the hinged jaw. Then we have another question that says, do we have legs? Then we go on to fish and other organisms. Then we answer these questions all along the way. So the question is this portion right here. So this taxa, we can form groups, but we need to name these groups. So back in the 1700s, a researcher, he's a botanist by the name of Carl Linnaeus, a Swedish botanist, came up with this plan for naming groups, and we go from the most diverse group down to the most specific group. And a most specific group is called the species. And we can then further divide that possibly even to a subspecies. So the science of organizing individuals or groups into these taxa is called taxonomy. So creating the taxa, and the naming system of what these groups were called came up by Carl Linnaeus. So we start from a root, and we start breaking groups down. So here we're seeing only one pathway. But if we start with all eukaryotes, then we have other kingdoms which would come off, but we have kingdom Animalia. Then from the kingdom, we can further break down into a phylum. Then from phylum, we go to class, class to order, order to family, genus to species. So it's not until we get to the species where all of the organisms within this species are able to reproduce, to mate, reproduce, and produce individuals that are both alive and themselves fertile. So I will ask you to know these categories: domain, kingdom, phylum, class, order, family, genus, species. And there's a number of different rhyming schemes that you can use to help you remember them, but it's important to know which is the most diverse. So the most organisms would be in this group. And then as we go down the path, we're going to get into smaller and smaller subgroups. So within kingdom Animalia, there'll be a number of different phyla. Within each phyla, there'll be a number of different classes and a number of different orders, etc. So when we get down to the naming, well, the names are in Latin, and the best way to use the most common form of naming is something called binomial nomenclature. Nomen meaning name, binomial meaning two names. And this is what we give to each species. Now, for each species, we could go back all the way to domain and tell their domain, their kingdom, their phylum, etc. But that would be, rather cumbersome every time you wanted to use their scientific name. So in general, we just use, when we talk about a species, their genus and their species names. So in writing those, we always write the genus with a capital letter to start, and then the entire name is also italicized, including the species name. So the species name is not capitalized. So if we see some examples here, we see Homo sapiens, that's us. Homo is the genus, sapiens is the species, and we see that the naming is in italics with a capital H for the genus and a small s for the species. Now, if you're not typing, if you're handwriting, or it's more easier, you can instead underline instead of using the italics, but you need to do one or the other. Now, within some species, we have a subspecies. So here we see Canis lupus familiaris. These are domestic dogs. So Canis is the genus, lupus is the species, and this is the subspecies. Now there are, of course, all the names that come before the genus and the species. Those names are capitalized, but not italicized. So here are some examples for different types of taxa above. And we see that Homidae is a family, primates is an order, and Mammalia is a class. Those are just some examples. Now at this point, you don't need to know what Homidae is, but you do need to know the order of class, order, family, genus, and species. So we have this naming scheme. How do we know how to put certain individuals into certain groups? Well, we need to look for evidence. Evidence to determine evolutionary relationships that organisms actually belong in the same group. So we use lots of different pieces of evidence to support this. We can use morphologic or anatomy. We can get that through fossils or through living organisms, physiologic or function, behavioral, and lastly genetic, which would be molecular, going right to the DNA or the RNA. So here we have the morphological. We are looking at the structures inside organisms to see how similar they are. These are called homologous structures. Homologous tells us that homo means same. They have the same kinds of structures, even though they might look slightly different as we go through different organisms. So we know that we are able to put these organisms all into the same class of kingdom Animalia, and then we will be able to subdivide them once we go beyond that. So we see that due to a similar evolutionary origin, they have the same common ancestor. We can also then go and look at genetics or developmental origin, but here we're looking at the complexity of the character to determine, did it come from the same ancestor? And keep in mind that while structures might look similar, they may not always be similar. So the underlying bones of vertebrate wings are homologous, common origin, but not the flight surface of the wing. So the bones are the same, but then the material beyond that is not the same. Now, again, looking at morphology still, looking at the structures, we have to be careful because we might have something that's called analogous structures. Analogous means they do the same job, but we can't closely tie birds and insects together. Even though they're both animals, these are vertebrates, these are invertebrates. So they have a similar functional structures, but they're not going to be in the same classes, etc. So we see that due to evolutionary convergence, convergence is where you have taxa or groups of organisms that aren't related, but they develop similar structures because of their environment. So it was beneficial for birds to fly, and it was beneficial for insects to fly. So separate from one another, they both evolved wings in their species to adapt to their particular environments. But they do not have a close common ancestor. And then we move on to molecular comparisons. So this is the most modern pieces of data or evidence. And so here we're actually looking at DNA sequences and how close are they. And from that, we can really find some interesting evolutionary relationships looking at the DNA. Now, keep in mind that there are still some limitations. There's only those 4 different nucleotides in DNA, A, T, G, C, and sequences might look the same when in fact they aren't. Also, when mutations happen, remember we talked about that last semester, things like insertions where a new nucleotide is added, deletions where one is lost, or frame shifting, that the order of the sequences could look like they are the same between different species, and they could look the same and may not be, and then they could look unrelated and actually be the same. So why does it matter to do this mapping, to do this grouping, to find out which species are related to one another? Well, tops on the list, it's very important to understand evolutionary processes, how these species evolved back from common ancestors. But additionally, knowing these relationships could help us understand issues about human health and medical concerns. Helps us to track evolution of parasites and viruses. We did this with the COVID-19 virus just this past year. We can use biotechnology if we know relationships, evolutionary relationships, to produce more productive crops and domestic animals. We can use it to make better drugs. An example is human insulin. And we certainly can use these to help threatened and endangered species in efforts to preserve them going forward. So lastly, I want to go back to Charles Darwin, who gets the credit for starting our modern understanding of evolution. And it's interesting to note that in his writings, he actually drew out phylogenetic trees. So we can go back and look at his original manuscripts and find these phylogenetic interrelationships. He says, I think this is how it happened. Since then, of course, we have lots of new technologies that have changed the way we think about evolution, and we're different from in the past. For example, in the past, we used to think fungi and plants were fairly closely related. Now we know that fungi are more related to animals than they are to plants, based on a lot of different evidence. We also used to think that birds were a separate class of vertebrates called class Aves. Now we know that they came from a reptile common ancestor. So that's changed the way we think about that form of phylogeny. Thank you very much.